During the cultivation period, it is necessary to take samples periodically for cell count and microscopic observation. It is also highly desirable to be able to preserve these samples from day to day, so that samples from different days can be compared for morphology. In our laboratory, we prefer to use 24-well plates to prepare samples for microscopic observation. The amount of sample required for microscopic observation, of course, is dependent on the concentration of microcarrier used. Too large a sample results in thick layers of microcarriers and interferes with the viewing of cells. Observation should be made without staining first. This allows one to detect unattached cells before they are removed in the fixing and staining steps. In most cases, a healthy microcarrier culture should not have many cells in the suspension. The fixing and staining procedure is usually combined into one step using a fixative staining solution. Drops of the staining solution are added to the well. The suspension is allowed to sit for a few seconds before PBS is added. A short period is necessary to allow the microcarriers to settle to the bottom before suction is applied to remove approximately 70% of the solution from the well. In removing the solution from the well, it is easier to apply pipette tips to the wall of the well so that the amount of microcarrier being sucked off is minimized. Most of the dead cells falling off from the microcarriers are spherical in shape. When excessive mechanical force is used, you may see elongated cells with one end clinging onto the microcarrier and the other end in the suspension. Another important phenomenon to look for is the cell distribution on microcarriers. A good microcarrier culture should have a relatively narrow cell distribution on microcarriers. One should not have a population of heavily populated beads and another population of sparsely populated or even bare beads. If this has occurred, in general, the inoculation procedure has to be improved.